We're going to finish talking about Mendel and basic Mendelian rules, and then we'll talk about why Mendel's rules are true with the view of chromosomes or cells undergoing division in meiosis. So how chromosomes are being segregated to make eggs and sperm. So basically we're tracing through Mendel's laws and why they're true biologically speaking. Okay, because Mendel started off and he just postulated his rules. He postulated his rules not knowing the biology behind these things, right? I mean, people were starting to do microscopy and looking at cells, but we didn't know anything about DNA, hereditary material, how cells divided. So Mendel just kind of created these, much like Darwin's view of gemmules. His little particles, his particulate theory, was just kind of an abstract theory. It was a way to make the numbers of the offspring work, okay? So we'll talk about his two laws here, and then we'll talk about what we now call classical genetics, the idea of Mendel's laws being applied to the truths of cells dividing in meiosis, okay? So Mendel took his particulate theory, and his particulate theory, kind of out of it came two laws, two requirements that is, two predictions that his theory makes. The first law we call the law of segregation, and he said each animal contains two particles, and when it makes gametes, only one of the two particles is inherited, okay? So gametes are inherited, or inherit one of the two alleles. And those two alleles do not blend, right? He says particles can either be dominant or recessive, but there's no blending intermediates, right? Either one trait is dominant and the other is recessive, or vice versa, okay? So this is in stark contrast to Darwin's gemmual theory, because Darwin said, well, it's a numbers game. How many particles you acquire of one type or the other is what's going to give you the ratio of blending, right? So if you get 90% black particles for black skin color or black hair color, and you only get 10% of the particles, then you're going to be a grayish, but grayish more biased towards black, right? So the gemmual theory says blending and proportional blending, right? Mendel says no blending at all. You only get two particles. There's no statistics game. It's either you're dominant or you're recessive, okay? His second law is the law of independent assortment. This basically just states that if you're looking at, and he's looking in peas, so we use pea plant experiments. If you're looking at the flower color is either going to be dominant purple or recessive white. And it does not matter what shape the little seeds are. It says you just think about traits independent of each other, and that's where his theory works. Okay, so if you're just thinking about flower color, then this is then his mechanism is going to work, irregardless of whether you've got wrinkled seeds or not. Okay, so the alleles of one gene, and we're using modern language, but we could say the particles of one character don't affect the particles of a different character. Okay. This is kind of preempting the idea of chromosomes, right? If these particles, if these alleles are separated by chromosomes, then chromosomes don't interact with each other, and so they're going to be independently assorted, okay? Is this clear how his particle theory, how these two laws have to be true based on his particle theory? Okay, so let's look at his theory now superimposed on what we know about cells, okay? So the classical genetics, or sometimes called the Mendelian chromosome theory of inheritance, says that these particles are actually genes, DNA sequences located on chromosomes, okay? So genes are located on chromosomes. That is the region that codes for a certain character, like flower color or seed shape, and that these are located on the chromosomes. We call the position on them loci. So where a gene is located on a chromosome is called a loci. And we know that the independent assortment is true because loci are located on separate chromosomes, and that these chromosomes are segregated when a cell divides. So if a cell is going to divide into gametes, it's going to divide through meiosis. And those two chromosomes with those two different traits, those two different particles, according to Mendel's terms, are going to separate when you make eggs and sperm. Okay. So here is, let's think about our smooth and wrinkled seed shape. Okay, so here's smooth, pure breeding smooth, pure breeding wrinkled. If we cross these two, and this is the Punnett square, or the Bateson square, if you want to give credit. A smooth individual has two particles. They're both the dominant ones, so big S's. The wrinkled, this is a pure breeding strain, a pure breeding individual, two recessive small s's. And so in the F1 generation, everyone is a heterozygote. They're inheriting one of the particles. You know, the only particle you can get is a big S from this smoothed individual. You can only get a small recessive particle from the wrinkled. So everyone is smooth because they've got a dominant one, and they're all heterozygotes. Okay. If you think about the gametes that this individual can make, though, it now has a choice, right? You can either give a dominant or a recessive. And this is illustrated here in a dividing cell. Okay? This is a cell. It's either an oogonia. An oogonia is a cell that is destined to divide. When it divides, it's going to become eggs. Spermatogonia is a cell that's going to, when divide, become sperm. Okay, so either in oogonia or spermatogonia, you're going to undergo meiosis. So in meiosis, you go through, this is the genotype of one of these individuals. Okay, heterozygous, came from two pure breeding strains. I've got a big S on one of the chromosomes, and here this red chromosome we'll say is the maternal, came from the mother's side, or the ovule. And the S came from the father's side, or the pollen contributed that allele, okay? As you go through meiosis, and hopefully this is a review, in past years I've gone back through mitosis. I'm assuming you guys have a good handle on mitosis. If you're fuzzy on the differences between mitosis and meiosis, go back to chapter 12 and review yourself. I'm just going to talk about meiosis because I'm more interested in sex and reproduction here, generations, inheritance over generations. There's interesting things that happen in mitosis, but we're just going to stick to eggs and sperm right now. Okay. So first phase of meiosis, it's going to go through S phase, and it's going to duplicate both of these genomes, or both of these chromosomes. Okay. So we're showing you only one of the many chromosomes that are in pea plants, but just for simplicity's sake, we're just going to think about these two. Okay. So S phase, you replicate the two of them. So for a homozygous, or I'm sorry, for a heterozygous individual like this, its parents are down here, right? This is a smooth parent, the female, she's got two big S's. In meiosis, you separate those two. These are now going to become your eggs or sperm, right? In the male, these are going to segregate, and you're going to be able to pass on one or the other. When egg and sperm come back together, now I've got a heterozygote, okay? So now I'm going to walk through the different crosses, and on top of the genotypes, I'm going to be showing you the actual chromosomes that are being inherited here, okay? So, smooth seed. This is so we created the F1 generation. We took those two pure breeding lines, we created the F1, they're heterozygotes. So here's a female heterozygote, a male heterozygote, and now they've got the option, right, to give a big allele or a dominant allele or recessive allele, dominant or recessive. Okay, so when I cross these, I do another Punnett square, and you can inherit either two of the dominant alleles, you'll be smooth. You could inherit a dominant allele from the female, so female is on this side, dominant allele from the female, a recessive from the male. You could be smooth, but a heterozygote. There's another way to do that. To get the same phenotype, you'd have the smooth dominant allele from the father, the recessive wrinkled from the mother, but we're a heterozygote, you're going to be smooth. Okay? Two chromosomes with both of the recessives going to give you the wrinkled phenotype. Okay? So in this monohybrid cross, we call it a monohybrid because we're talking about one gene and we're crossing heterozygotes or hybrid individuals. Right? This individual is a hybrid because it's got one of each of the two alleles. Right? So one gene, heterozygotes. And this is the traditional 1/4 of the genotypes are going to be dominant, homozygous dominant, right? Both alleles for the dominant. You get 2 out of the 4 are going to be heterozygous, and 1 out of the 4 is going to be homozygous recessive. Homozygous meaning 2 of the same alleles. So 2 of the same alleles for dominant, homozygous dominant, 2 of the same alleles for the recessive, homozygous recessive. Yeah?
So what's meant by monohybrid? Is heterozygous?
We're just talking about one trait, one gene, and that's the gene that codes for your seed shape, right? That's the mono, right?
Okay.
One trait. Hybrid means we're crossing 2 individuals that are heterozygotes. So each of those people in the cross are hybrids. They've got 1 of each. They're heterozygotes. So when I say heterozygote or hybrids, that's meaning the same thing right now. Okay. So this gives you your standard, the phenotypes. We talked about the genotypes. The phenotypes then are 3 of the 4 are smooth. Now underlying that is 2 different genotypes, but phenotypically what the things actually look like are smooth. 1 out of 4 is wrinkled. This is Mendel's 3-to-1 ratio. 3 out of 4 are smooth, 1 out of 4 is wrinkled. Okay? This is monohybrid. Now you can apply Mendel's rules. Mendel's second law says independent assortment, right? How things are inherited based on your seed shape is completely independent of how things are being inherited based on your flower color, right? So here, instead of flower color, we're also overlaying on top of this seed color. So seeds can be yellow or green. They can be smooth or wrinkled, right? So basically, I'm just adding a second layer of Mendel's laws right on top of the first one, okay? So here I have a pure breeding smooth and yellow. Yellow is the dominant. Green is the recessive here. So homozygous for smooth, homozygous for yellow. The only gametes this individual can give is 1 smooth chromosome and 1 yellow chromosome. Okay, we're complicating this a little bit because now I'm showing you 2 chromosomes. We could say this is chromosome 1, this is chromosome 2. Okay, they've got 2 copies of each because this is a diploid organism. So for every chromosome, they've got 2 copies. But when it makes an egg or sperm, this is a female, so it's making the ovule, only giving 1 of each of the chromosomes. Okay, particulate theory. So this is a pure breeding wrinkled green. So wrinkled is small s, green is small y. The only thing this can give is that. So the first generation is now what we call dihybrid. Okay? Because at both traits, they're hybridized. So all the offspring that come from this are going to be heterozygote for smooth, or for shape, heterozygous for color. But underlying them now are those recessives. And this is where you're going to get the diversity that comes back out. Okay? So if I did a dihybrid cross, that is, I crossed 2 individuals who are heterozygous at 2 traits, 2 loci, now things start getting complicated, right? Because now I have to interweave all of these different phenotypes, right? You could either be, and this is hard to see. Just look at the color and the shape right now. You could be yellow and smooth. All of these guys are yellow and smooth, yellow and smooth. This one's yellow and smooth, yellow and smooth. You could be yellowed and wrinkled. That's these 2 guys. You could be green and smooth, these 3. Or you could be green and wrinkled, that 1 individual. So I started off with just 2 different traits, but now if I start mixing them, I actually have 10 different phenotypes now. Or I'm sorry, no. Let's see. 4 different phenotypes. I'm jumping ahead to the next trihybrid cross. You get 4 different phenotypes, right? Yellow and smooth, yellow and wrinkled, green and smooth, green and wrinkled, okay? And these, Mendel did these kind of experiments, dihybrid crosses, and he got a typical phenotypic ratio of 9 to 3 to 3 to 1. So 9 of them are going to show both dominant traits. dominantly smooth and dominantly yellow. The hybrids, the ones in between that showed one dominant trait and the other recessive, one of those is going to be 3 out of this, all of the individuals. So that would be something like one of the dominants here is dominant for smooth, but recessive for the green color. I get 3 individuals like that, smooth and green, 1, 2, 3. Or the alternative is to have be dominant for the other trait, dominant for being yellow, but recessive for the shape. So I've got dominant yellow, but wrinkled. Also 3 out of these individuals are that. You only see the one that has both recessive traits. You see that very, very rarely. And so that's only 1 out of all these 16 individuals gets that. So this is the 9 to 3 to 3 to 1. Typical of a dihybrid. This assumes that these are on different chromosomes, okay? Because in meiosis, the chromosomes are separating independently. You can just keep doing this. This is a trihybrid cross. At this point, back here, this was manageable. In a dihybrid cross, doing a big Punnett square like this is manageable, right? You just put all the possible gametes that one individual can make, all the possible gametes that the other one could make, and this is, I mean, dealing with 16 boxes is manageable. If you get to a trihybrid cross, though, this would start to be unmanageable, right? Because I mean, you could do it, and if you want to do it, go for it, but it's going to be a huge Punnett square, and there's going to be a ton of individuals to keep track of. It's probably easier to start doing a kind of flow chart method. So in a trihybrid cross, now we're going to throw in, you got seed color, seed shape, and now we're talking about flower color as well. And that's on a different chromosome. And actually, thankfully for Mendel, these were on separate chromosomes. In the pea plant, these are on separate chromosomes, and so they independently assort. If these had been on the same chromosome, it would start throwing his numbers off, and we'll talk about that. But in trihybrid, it's going to get more complex. So yeah.
So if they're on a different chromosome, so like he has there, smooth yellow and purple, if those are each on a different chromosome, would you just cross each of the components separately? You do your own individual Punnett square for smooth and wrinkled, your own Punnett square for yellow and green?
Well, yeah, you could if you were just thinking about that one trait. But if I want to think about if I took a smooth yellow purple plant and crossed it with a wrinkled green white plant, what are the possible offspring that can come out of it? I have to think about all 3 traits at the same time.
Okay.
But Mendel says the individual laws of inheritance hold true for each individual one. So if I looked at them in isolation, if I just looked at purple, it doesn't matter what the other traits are. 3 to 1 ratio is going to hold true. In a trihybrid, though, we say, okay, well, we've got 3 traits now. How are those going to come out? What are the ratios going to be if I put all 3 of them together? And basically, they're just like an exponential difference. So first we had 3 to 1 and with the monohybrid. Then we did a dihybrid, we had 9 to 3 to 3 to 1, right? So you had 3 instances of 3 for 9, 2 instances of 3, and then 1. Now we have, in a trihybrid cross, and you could do this, and I think I assigned you a question to do this at one point. You're going to have now, since we're talking about 3 traits, you've got now 8, 1, 2, 3, 4, 5, 6, 7, yeah, 8 possible phenotypes. And they come out at a characteristic 27 to 9 to 3 to 3 to 3 to 1. I'm sorry, 27 to 9 to 9 to 3 to 3 to 1. See how they're kind of, there's a name for this mathematical expansion. It's not exponential, but it's, yeah, I forget the name of it. There's some statistic rule for this. If somebody knew math better than I, they could tell you. But basically, we're just raising each one of them to the next power. So 27 are going to be dominant for all traits. 9 of them are going to be dominant for 2, recessive for 1. Dominant for 2, recessive for the other one, dominant for the other 2, recessive for the third one. This 3 is going to be recessive for 2, dominant for 1, And then there's 3 ways you could do that. And then only one out of all of these is going to actually be recessive for all 3 traits. So when you're doing pedigree analysis, when you're thinking about recessives, you should always be thinking they're going to be the more rare instances. In your homework assignment, there was like some pedigrees, and it said estimate which one's being inherited based on a recessive or dominant mechanism. If the trait is rare, probably your first guess should be it's probably recessive, okay? Because recessives just tend to kind of fall in the background, right? So when you're trying to sort out these, I would do a flow chart. So I'd say start with one trait, smooth or wrinkled. Okay, if I'm talking about a smooth, is that yellow or green? If I'm talking about a smooth yellow, is that one purple or white? That will help you determine your genotypic and phenotypic ratios. If you just do it in a flow chart, it's going to be easier than putting that whole grid together. So yeah.
Can you just do 2 to the exponent of how many traits there are in order to determine the number of phenotypes? There's 2 alleles for each trait, and then 2 to the power 3 is 8.
Yes, I think that always holds true. Yeah.
So you could do that.
Yeah, if there's no linkage, if they're on separate chromosomes, that will hold true. Yeah. Okay. So Mendel's laws explained all of his instances of phenotypes in pea plants, right? And it was almost kind of serendipitous because the traits he looked at were all in different chromosomes, so they all segregated independently, assorted independently, and so he could explain a lot. Now, there are limitations to Mendel's laws, and we'll get into how chromosomal theory can still explain them, but you start deviating from Mendel's laws, okay? So some of these are, some traits do appear to blend. Skin color in humans appears to blend, right? If you have a dark-skinned person and a light-skinned person, you don't have 3-to-1 ratio, 3 dark-skinned children and 1 really light-skinned, right? Some things actually do seem to blend. You usually get all the offspring or some kind of intermediate between them, right? I don't know, phenotypic things are, facial features like, nose shape and ear size and, I don't know, a lot of traits like these, eye color, don't appear to be like quite dominant recessive like Mendel's laws predict them. Some traits don't even appear to conform to Mendelian ratios at all. Some traits, dominant recessive or blending, doesn't quite seem to be the case. You have things that run in families, but there's no real way to predict when it's going to show up or when it's not going to show up. Things like disorders, diabetes, twinning, heart disease. These things seem to kind of run in families, but they don't map onto any chromosomal theory or Mendelian ratios. So yeah.
When they say that something skips a generation, are they talking about just the ratio is greater in the next generation to have that trait?
Well, I mean, there's 2 ways for that term to be used. Most of the time, it's people kind of talking casually about things coming back in their family, and so they're like, oh, it skipped a generation. But it's usually kind of colloquialism, and we're not really talking specifically. Yeah. I mean, if you're talking specifically about pedigree analysis, if something doesn't show up in one generation, but then shows up in another, but then is hidden and then shows up, that would be true skipping of generations. And there's few traits that actually do that robustly, right? A recessive allele may be rare, but it might not always just skip generations. It might just be rare in each generation. So yeah, that's not usually a term that geneticists use. Yeah. Yeah, I mean, we hear a lot of things all the time that are not true. So, yeah. Like, he has his father's nose. It's not true at all. So things like cancers, some cancers you can predict, and they act in dominant recessive, but usually not. It's usually kind of an environmental thing too, and there's a lot of more complicated genetics around cancer. Later in the semester, we'll talk about some of the ways that you can actually inherit specific types of cancer, but most of the time you inherit a propensity for cancer. You have a higher likelihood of having cancer. It's not like 3 of the children are going to have cancer and 1 is not, right? So heart disease, all kinds of neurological disorders like Alzheimer's, Parkinson's disease, These things do seem to have a genetic component to them, but they're not following Mendel's laws. So in the next couple of lectures, we'll talk about deviations from Mendel, so ways we can still predict but that don't follow Mendel's laws. And then we're going to start getting into stuff that doesn't actually map onto any mechanism at all. All right. So I want to talk about meiosis a little bit and talk about gametogenesis, making eggs and sperm. And this will set us up to start talking about when traits are sitting on the same chromosome. Okay? So Mendel doesn't know, his laws don't know how to deal with traits that are on the same chromosome. He didn't even know there were chromosomes. We have the ability to know that they can sit on chromosomes, or they do sit on chromosomes, right? And so I want to talk about how you make eggs and sperm, and talk about recombination that happens in meiosis. This is where chromosomes are actually going to swap information. And so that will then help us to think about these beyond Mendelian ratios. When we start talking about linkage, deviations from Mendel's ideal laws, right? Ideally, in a perfect world, every gene would be on a separate chromosome and it would independently assort. But they're all stuck together, so we have to think about that. All right, so this should be a bit of a review. We'll just walk through the stages of meiosis. We'll talk a little bit about gametogenesis and hint in a little bit about kind of developmental biology, and that'll set us up for next time talking about linkage. All right? So here is a cell undergoing meiosis. The cell has already gone through the G1, the S phase, and G2. Right? So the cell is in sufficient size, it's got sufficient number of organelles, it's polymerized all of its microtubule, or it's made all of its microtubules, it's duplicated its genome, it's ready to separate the chromosomes. Okay? So here is a cell in early prophase 1. Now meiosis has 2 phases, right? 2 cell divisions. In prophase 1, the nuclear envelope starts to break down. The chromosomes are beginning to condense, and so instead of just a mass of chromatin, euchromatin, and heterochromatin, the chromosomes are actually condensing down so much that you can actually start seeing individual chromosomal strands. Okay? In mid-prophase, we're condensed even more. And in meiosis, this is the interesting that happens, is the maternal and paternal, the mother and father's version of the 2 chromosomes, start to find each other in the cell. Okay? So they've already duplicated, so there's 2 copies of mom's chromosome 1, 2 copies of dad's chromosome 1. And in the cell, those 2, what we call homologous chromosomes, because they're both chromosome 1, and they both have, by and large, the same sequence, but they differ slightly, right? They got mom's versions and dad's versions, different alleles of each gene. Those 2 homologous chromosomes start to find each other in the mid-prophase. We call those pairing ups tetrads, because there's 4 chromosomes now, 2 copies of mom, 2 copies of dad. So here are the 2 copies of mom, you can't distinguish them yet, but here's 2 copies of dad. They start finding each other and pairing up. This is an interesting phenomenon, and it's not like the chromosomes themselves are finding each other, right? This is all mediated by proteins that are doing this. The proteins are grabbing the homologous chromosomes, bringing them close together, and they're going to encourage this, this behavior. So this is particular genes that get expressed in just the oocytes or the oogonia or spermatogonia. Okay. In pro-metaphase, the chromosomes start moving towards the middle of the cell. So they're starting to hook up with the microtubules. The microtubules are pushing them to the center. And this is where recombination starts happening. And that's where the maternal chromosomes and the paternal chromosomes have the opportunity, they don't always, but they have the opportunity of actually breaking the chromosomal strands and swapping arms. This is what's going to give you greater genetic diversity, right? If this didn't happen, then any trait you had on a chromosome would always go with that other trait, right? So if, let's think about the pea plant. If purple flower color went with smooth seed shape and they were on the same chromosome, those 2 traits would always go together since they're on the same chromosome. In our typical separating of chromosomes and the particulate theory, those should always have to go together. But recombination is going to make it possible for those to be separated out from each other. So instead of making just partial clones every time an organism makes eggs and sperm, basically they have the opportunity to like reshuffle their entire genome, right? Every trait in theory has the possibility of going with another trait. So it might be more or less common, but at least they do have the opportunity. This is why you see such genetic diversity, such organismal diversity in populations. It's because they can go either way. Yeah?
Just makes like a question. I just want to clarify. So it's
Yeah.
always, so the chromosomes line up so that the traits, so like the actual, the alleles themselves will swap for the same trait. So you won't ever get like this trait goes over here, but we didn't get a swap back. So
Well, yeah, you always get a swap back. We'll talk about this in a little bit more detail here, and I think it'll clarify this question, but you really are going to recombine and swap, okay? If you don't swap properly, it's going to stop, and you're probably not going to make an egg or a sperm, okay? So let's zoom in on that recombination event. So here is a tetrad. This is actually a scanning electron micrograph of two chromosomes coming together. So here is, these are identical chromosomes. We'll just say that the top one is the maternal, the mother's version, and the bottom one is the paternal. So this cell has undergone S phase. It's duplicated its chromosomes. So now I have two maternals and two paternals. This dark region is the centromere. That's where they have that centromeric repetitive region, highly condensed, and that's where the two chromosomes, and we call these two chromosomes sister chromatids. And they are identical in sequence. And they just came through S phase, the cell just replicated them. So these guys are identical in terms of their genetic content, held together by the centrosomes. Same with the paternals, two identical copies held together at their centrosomes. And here, at this tetrad, there are two recombination events. The maternal and the paternal are recombining here, and they're also recombining here. Again, them finding each other is not random, right? Proteins in the cell are bringing them together, and proteins in the cell are actually inducing this crossing over. This is called homologous recombination, because the homologous chromosomes are the maternal and the paternal, right? So if I'm comparing these two to each other, we say that they're homologous. That means they're similar but not identical. Sister chromatids are completely identical, right? So these are sister chromatids, these are sister chromatids. Any one of these to any one of these compared are homologous, okay? They're both chromosome 1, but they're slightly different versions, so we say that they're homologous. Actually, this illustration is nice because it's going to show you that it's not just two of the chromosomes that are the ones that can recombine. Any one of the maternals can cross over with either one of the paternals, and vice versa. So if you could actually go in, and so here's a cartoon illustration showing you exactly which strands are recombining. Here we have the lower mothers, the lower maternal, is crossing over with the upper paternal. So we're having a crossing over event right here, and those are going to switch information, okay? If you keep looking, though, along that mother's chromosome, so it's going to cross over here, but if you keep looking at the red, it's now crossing over with the other paternal chromosome. And so this can happen any one of the 4 different ways. Okay, so it's not just one of the strands doing it. It's both of the strands at multiple places with the potential to cross over to any one of the other 2. Okay, so yeah.
But there's not a difference if it actually coming at the end with the upper or the lower.
I'm sorry, say that again. Say it louder.
So there's not a difference if it's actually like at the end where the maternal is pairing with the paternal.
Yeah.
Like whether it's like pairing with the upper one or the lower one is still the same because they're the exact copy.
Yeah, because they're the same sequence, right? But what you should see, though, is one of these maternals didn't cross over at all. So it's holding all of the same alleles that the mother had, right? So if I make, if this is an egg, an oocyte dividing to become an egg, if that egg takes that chromosome, it's going to have the exact same traits that the mother had, right? If the egg is made from and uses this chromosome, it's got, well, this region comes from the mom, this region comes from the mom, but it's going to cross over and it's going to get this region from the dad, just that entire length. That's different than not crossing over at all. If we use this chromosome, it's going to have this from the father, this from the father, this from the mother, but then back to the father. So every single one of these chromosomes is now a unique reshuffling, okay? Even though it only has two options, it can either reshuffle with moms or with dads, how many times it uses moms or dads is variable. Does that make sense? So, does it? Yes, okay. You're looking like you're still confused, but maybe there are people that are still confused, yeah.
How do you say the word chiasmata?
Chiasmata.
Chiasmata?
Yeah.
Is it the point where they cross over each other, or is it the space that's being...
It's the point at which the two of them are crossing over. So right where it makes, it's just a description of what it looks like. So it looks like the Greek letter chi or an X. So it says the place on the chromosome that looks like an X. Now what we actually mean is that exact location where the genetic information is going to get swapped. Okay. So yeah.
So recombination only occurs in prometaphase. Is that considered part of the latter half of prophase 1?
It's like the early part of prophase 1.
Okay.
Yeah, yeah. I'm sorry, early part of meiosis 1, right?
Yeah, but would you consider prometaphase as prophase 1?
You know, this is a continuous thing that's happening, and we've kind of put arbitrary labels on when it's pro-metaphase and when it's, you know, has gone to a different phase. This is a dynamic event, and we're just trying to demarcate when things happen. So it's convenient terminology for us. I don't so much care that you know when, if it's in, you know, pro-metaphase or late metaphase or early metaphase, or as long as you know the order of the events, I don't care that you like know the terminology. So yeah.
I'm just wondering if there's any like proteins involved that actually help them cross and stuff.
Thank you for asking. There are proteins that are called recombinases. These are the proteins that are actually inducing the chromosomes to break and to recombine. Okay? So the chromosomes themselves are not doing this. It's these recombinase proteins. In humans, one of these is called DMC1. There's many of these. They're called different names in different organisms. And we generally think of recombination as kind of happening randomly, but it really does seem to be specific to certain regions of the chromosome. And there's recombination maps of the chromosomes. So if you're interested in human chromosome number 1, you can get analyses of all of the different recombination events that we've documented on human chromosome number 1. And there seems to be regions of the chromosome that are more likely to recombine than others. And that's probably something to do with this protein's specificity for a segment on the genome. So there's probably places where, you know, recombinases like to sit down and sit down more often, and so that is a high point of recombination. And there's other places where it's more rare. There's interesting maps and interesting things that go about this. So in an ideal world, everything would recombine at the same rate, but it tends to have more instances here or there. So some traits will be higher linked than others will be. Okay, so these recombinases bind in clumps to the two strands. So right here, recombinases are going to be clumping there. And that's what actually induces the DNA strands to denature. Because remember, these are really condensed chromosomes, like highly condensed around their chromatin. And so if you're going to get actual breaking of the DNA strands and recombining, we're going to have to denature that. So these guys denature, so they displace some of these histones and all the scaffolding proteins, expose the DNA strands, and so then you can actually cut them and recombine them. Okay? So they denature the DNA strands, you actually cut the chromosomes, and then they reanneal reform the phosphodiester bonds with the other strand. Okay. I'm going to show you the holiday structure, and then I think we're going to take some time in lab to actually work through this a little bit. The holiday structure is now, we're looking at the double-stranded DNA now. Okay, so up until this point, and I'm sorry, it's just the way we have to do it. Right here, we're showing the chromosomes, right? So when I'm talking about this maternal chromosome, this is a double-stranded piece of DNA, but I'm just showing it as a line because it's really highly condensed chromatin, right? If we were to actually zoom in on that, though, it's double-stranded DNA, right? And so we have to talk about both strands of DNA breaking and recombining, okay? So here is that one strand of the mother, one chromosome from the mother, one of the sister chromatids. This is the homologous chromosome from the paternal side, one of the sister chromatids from the father. And the two lines are the double-stranded DNA, okay? So hydrogen bonding together. What actually happens when these recombinases clip the DNA and recombined is they first just cut one strand of each of the chromosomes, okay? So this lower strand on the mother's is going to get cut. The upper strand on the father's is going to get cut. Those are going to come apart, and they're going to cross over each other and reanneal to the other side. So if I was looking along here, if this was the 5' end of the paternal, I'd be moving along, and then right at the point it gets cut, I'm going to reanneal it over here, and so that's going to be now continuous, okay? If I was looking at the mother's, start here, I'd move along right to the cut point, and then I cross over to the other side of the, so just one strand is broken on each of the two. The other strand remains intact. And this is why we're going to go through it in lab, because you start to have to think three-dimensionally. To begin with, you cut those, so they're sitting next to each other. You cut them, and then they cross over and reanneal, right? So I've got this actual, arms crossing each other. What happens is those arms are crossing each other, and then the recombinases actually unfold it so that they weirdly kind of come apart. We're basically just flipping them over, and that's why this is any illustration. I've tried numerous illustrations, and none of them seem to sink in. So that's why we're going to spend some time in lab actually, like maybe using some thread or something and unfolding them. But what I want you to see is that top strand from the mother is this one right here, still intact, right? We haven't cut that phosphodiester bond in that strand yet. Here in the bottom, this is the bottom mother. Here it's here. That's the point we clipped it. That used to be right here. And now it's attached to the father strand, okay? The bottom father strand has not recombined yet. It's still intact, okay? So these are still hydrogen bonding together, but it's in this weird opened X shape, okay? I think it'll help if we actually put thread together and I show you how it twists open, okay? So.
I have a quick question. When we look at the whole entire chromosome,
yeah.
Look at like one chiasmata.
Yeah.
Is that just one holiday junction, or are there multiple? Just one.
Yeah. For every chiasmata, one holiday junction is happening.
In that whole entire
right. And the holiday junction is just it's just what it actually looks like at the two-stranded level. Okay. So I mean, in a sense, we're talking about the same location.
So like when we look at the chiasmata, even though it's a huge part of the chromosome, when you zoom in, it's actually just one.
It's actually one site, just one place that the two chromosomes are getting cut. Yeah. Okay. So we cut one strand, you form this weird, wacky intermediate, and what I want you to see is that we can resolve this and cut the other strand in two ways. We can either cut the maternal strand and the paternal strand. If we did that, if we cut vertically on this structure, then 2 copies of mom go with the other 2 sides, and sorry, my pointer's going out, other 2 sides of dad, right? That's what we typically think of as chromosome swapping, right? You cut both strands and reanneal them, both strands, to the father's chromosome. And on the other side, the father's chromosome would then both be cut and would link up with both mom's sides. But you could also resolve this way. That is, you could just recut the same strand that you just cut before and repair them up. So sometimes you cut the one strand, put it together, but then cut that same strand again and no real swapping happened. Only when you cut vertically are you really going to cross over. And then we'll talk about how this can slide back and forth, because if you notice, we're not cutting in the exact same spot. We've got this weird one strand maternal, one strand paternal overlap. Okay? But right now what I want you to get is that first one strand is clipped and re-annealed to the other side. And then the second strand is clipped and re-annealed to the other side. Okay? And it can happen, it can be clipped one of two ways. All right. I was skipping somebody's question for a while. Who was I ignoring? I forget. No? All right. Okay, so that's happening in the early to late, or mid to late late prophase, early metaphase, right? Once all the recombination has happened, then we actually enter into metaphase 1. So by the time all the chromosomes are lined up in metaphase 1, all of the recombination has either been completed or is nearing completion, right? Once all the recombination completes, then we go through anaphase 1. You separate out these now newly reshuffled chromatids, okay? So some of them may not have undergone recombination, some of them have, but we're going to separate out the homologous chromosomes, okay? We actually undergo cytokinesis. The cell divides in two. And then we just go into meiosis 2, the second round. And I'm not going to spend much time on this because, again, I'm going to assume that you know how to separate chromosomes in mitosis. The second phase of meiosis is almost identical to mitosis. Instead of lining up the homologous chromosomes, we just line up every single chromosome. So in metaphase, all of these now recombined. They're not sister chromatids anymore. Sister chromatids implies that the two strands are identical to each other. But now, and this is maybe a little bit hard to see, but some of it's red and some of it's blue, right? If the other one didn't recombine at all, then those aren't identical anymore. They've swapped some information, right? But we're going to now separate those two chromosomes from each other, okay? So chromosomes go to either side in anaphase, and this happens in both of the cells that we got from the first round, right? So first round of meiosis, we got 2 cells. The second round, we're going to divide those two, and now we get 4 cells. And now each of them have a unique reshuffling of every single chromosome, okay? Each of these 4 cells has now got 1 copy of each chromosome instead of 2, but each one is a reshuffling. So it's got a unique chromosome 1 and a unique chromosome 2, a unique reshuffling. Each of these is going to have a unique chromosome, a unique copy of every one of the chromosomes, a unique reshuffling. So you're never going to make the same egg or the same sperm twice, okay? So next time we'll talk about how this actually happens in the context of like sperm dividing, eggs dividing, but you've got the generic now idea of how the chromosomes go in meiosis.
Biola University offers a variety of biblically centered degree programs, ranging from business to ministry to the arts and sciences. Visit biola.edu to find out how Biola could make a difference in your life.